Good morning, students. Today, let me continue with this histology. And as far as today's class is concerned, let me explain you about the histology of spleen. As we observe in this slide, you will observe the structure of the spleen and its section showing the different histological details. In this, this spleen is a very important component of our lymphatic system. Our lymphatic system consists of so many components in it, like lymphatic nodules, lymphatic tissues, and some lymphatic organs. And in this picture, you will observe the details of the human lymphatic system, where there are several lymph nodes can be seen, and some lymphatic tissues are present, and some lymphatic organs are also shown here. Coming to the functions of our lymphatic system. Lymphatic system monitor the body surfaces and fluid compartments, example epidermis, mucosae and interstitium. It reacts to the presence of potentially harmful antigens recognized as non-self, and it controls autoimmune diseases like rheumatoid arthritis and type 1 diabetes, etc. The lymphatic system consists of A. Cells like lymphocytes, that is B and T lymphocytes, as well as natural killer cells. It also includes the antigen presenting cells like dendritic cells and Langerhans cells, that is pancreas, and macrophages present in different parts of our body. B. Lymphatic tissue, which is diffuse and nodular type. And C. Lymphatic organs like lymphatic nodes or the lymph nodes, spleen and thymus. D. Lymphatic vessels that carry the cells and fluid from one part to another part of the body. So basically our lymphatic system consists of 4 different components majorly, like cells, lymphatic tissues, lymphatic organs and lymphatic vessels. However, mucosae refers to lining tissue of the body cavities, example GI tract, that is gastrointestinal tract, respiratory tract and genitourinary tract. Some lymphatic organs are classified as primary lymphoid organs and secondary lymphoid organs. Primary lymphoid organs includes thymus, bone marrow, lymphatic nodules of the distal intestinal tract, that is ileum and appendix. And the secondary or effector lymphoid organs or tissues are as follows: spleen and lymph nodes, that is organs, and mucosal associated lymphoid tissue, which is popularly abbreviated as MALT. Example, the lymphocytes and lymphatic nodules in the lamina propria of GIT, that is gastrointestinal tract. So on the left hand side of this slide, you will be observing the different lymphatic organs like primary as well as secondary lymphatic organs, the details of which I already explained to you, and this diagram is for your reference. Coming to the structure of the spleen. As you observe here, the spleen consists of the outer capsule, and it consists of a number of inward projections called trabeculae, and inside the trabeculae you will observe a number of lymphatic nodules and the parenchyma tissue, and the major component is pulp, which is further subdivided into white pulp and red pulp, the details of which I will explain you in the later part of this discussion. And the spleen, as you know, it is a very important organ, and it receives a rich blood supply through arteries, and the impure blood is taken away from the spleen through veins. This is another diagram showing the structure of the spleen, that is anterior view and other histological details. So in the diagram of this spleen histology, you will observe that external it is covered out by capsule, and that capsule consisting of some fibrous connective tissue which produces some inward projections called trabeculae, and the trabeculae leads to the major part of this spleen, what is called as pulp. As I told you, it is further divided into red pulp and white pulp, and details are shown here in this diagram. And you will also observe this many projections here called splenic cords, wherein the splenic cords are the ones which carry away the the product produced from this spleen to different parts of the body. The spleen consists of a number of small spaces formed by the trabeculae which contain the splenic pulp, and the fibroelastic coat, the sheets of vessels and the trabeculae are composed of white and yellow elastic fibrous tissues. Spleen possesses a considerable amount of elasticity which allows of the very great variations in size, and small amount of non-striped muscular fibers are also present in the outer covering called cap. And the spleen is made up of red pulp and white pulp separated by the marginal zone. About 76 to 79% of a normal spleen is red pulp. This is what shown in the diagram. However, apart from this red pulp, there are some intermediary projections called white pulp. And as you observe in this picture, there is a very less amount of white pulp is there and more amount of red pulp. As the body is exposed to antigens and the immune system mounts an immune response in the form of antibody production, lymph nodules, that is germinal centers, appear in the white pulp of the spleen. So the germinal centers are shown here in this slide. Coming to the secondary follicle, which is shown here with the central artery cut in cross section, and note that the CA, that is central artery, has been pushed off to the side by the rapid expansion of the cells in the germinal center, which is abbreviated here as GC. So RP refers to red pulp, MZ refers to marginal zone, that is antigen presentation region, which is shown by dash circle, that is T cell rich in zone. Coming to the further details of the spleen histology. So as you observe in this diagram, there are some spaces in between these cords, what are called as sinuses, and sinuses are there in different parts of this section between the cords. Coming to the cells that are found in the red pulp, it includes B and T lymphocytes, erythrocytes, granulocytes, plasma cells, hematopoietic cells, plasmablasts that migrate from the follicles and the outer pulse after antigen specific differentiation, and it also includes macrophages. And there are some splenic cells which are large round cells. Amoeboid movement is shown by them and often contain pigment and red blood corpuscles in their interior side. And the cells of the reticulum may contain pigment granules in their cytoplasm. Coming to the white pulp. The white lymphoid follicles arrayed within the red pulp matrix. It contains lymphocytes and macrophages, dendritic cells, plasma cells, arterioles, and capillaries in reticular connective tissue network or meshwork. I have mentioned you about an abbreviation called PALS, that is nothing but peritoneal lymphatic sheets, that is PALS. Periarterial lymphatic sheets, which is abbreviated as pals. It is a lymphatic tissue sheet that surrounds the central artery. So pals is the lymphatic tissue sheet that surrounds the central artery, that is CA. Most of the T cells are of CD4 plus type. So in this picture you will observe that details of that pals component here in the white pulp region. Coming to the functions. Red pulp, it is mainly responsible for mechanical filtration of red blood cells, whereas white pulp, it is responsible for active immune response through humoral and cell mediated pathways. Humoral refers to the immunity which is offered through the body fluids, and cell mediated, as the name itself suggests, it is through some cells. And it is also responsible for the production of opsonins, properdin and tufsin. It is also responsible for creation of red blood cells, that is hematopoietic functions up until the 5th month of the gestation, and after birth erythropoietic functions cease except in some hematological disorders. It retains the ability to produce lymphocytes continuously. It is also functioning like a storage, as it contains approximately one-third of the total body platelets and a significant number of marginated neutrophils. And it is sequestered cells are available when needed to respond to bleeding or infection. And the normal human spleen does not sequester or store red blood cells. It is also responsible for the removal of red blood cells. It removes senescent and defective red blood cells. Senescent means which are young. And through a unique organization of its parenchyma and vasculature. After the normal lifespan of RBCs, that is about 120 days, they will be broken down and some parts are recycled, and it removes all abnormal red blood cells also. So this destruction of this RBCs, the old and or not RBCs, that is one of the major function of this spleen. And it is known for the removal of antibody coated bacteria and antibody coated blood cells from the circulation. And it also harbors some phagocytes, that is reticular cells and free macrophages of red pulp, and the modified reticular cells of the ellipsoid, and the free macrophages and endothelial cells of the venous sinusoids. And it also includes surface reticular cells of the lymphatic follicle. It is known for assisting the host in adapting to its hostile environment, that is, whenever there is an infection. And it performs the following functions: clearance of bacteria and particulates from the blood, generation of immune responses to certain pathogens, the generation of cellular components of the blood under circumstances in which the marrow is unable to meet the needs, that is extramedullary hematopoiesis. And it also known to monitor the antigens in the blood and the proliferation of lymphocytes, production of humoral antibodies. So these are some immune functions of the spleen. And the hematopoietic functions of spleen are as follows: formation of blood cells in the fetal life, removal and destruction of RBCs and platelets, retrieval of iron from RBCs, that is hemoglobin, and storage of RBCs and platelets, more so in non-human species, but very less is known in human species. The details of histology of mammalian spleen are as follows. Some important features. First one, spleen is a soft, highly vascular, blood filtering and the largest lymphoid organ of the body. The human spleen is situated in the upper left side of the abdominal cavity, behind the stomach and just below the diaphragm, behind 9th, 10th and 11th ribs. It is about 5 inches long, 3 inches wide and 1 inch thickness it has. And its indented medial border is called hilus, the details of which you have already seen in the second slide. Through the hilus, splenic artery enters and supplies to splenic tissue. And the spleen is externally covered by an outer serous coat, that is peritoneum, and inner capsule. Trabeculae extend inwards from the capsule and divide the parenchyma or the splenic pulp into incomplete compartments, and the trabeculae provide internal support for the spleen and carry blood vessels to spleen. Capsule, trabeculae and trabecular network contains collagen fibers, elastic fibers, smooth muscle fibers and reticular cells. So these are the components of the capsule and trabeculae. The splenic artery enters the spleen at the hilum, runs through the trabeculae for some distance, which is already shown in one of the previous slides, and then it enters the splenic pulp or parenchyma and branches out profusely. They are called central arteries or pulp arteries. So this is the one more diagrammatic view of this spleen histology. So as you observe here, there will be a capsule outer covering and that will produce these inward projections called trabeculae, and the trabeculae divides the parenchyma or the substance of the spleen into white pulp and red pulp, which is shown clearly in this picture. And it consists of a vein called splenic vein and splenic artery. As I said in the previous slide, that splenic artery, after it enters the spleen, it produces profuse branchings or many number of branchings, between which you can observe the presence of these venous sinuses and arterial capillaries, as well as central artery, which is shown in the one of the previous slides, and there will be the presence of red and white pulp. Splenic pulp, that is splenic parenchyma. The splenic pulp is composed of reticular tissue, lymphocytes, red blood corpuscles and macrophages. The splenic pulp is differentiated into two components, the white pulp and the red pulp. Coming to white pulp. The white pulp is characterized by aggregation of lymphatic nodules or splenic nodules enveloped around the central arteries. The central artery or splenic arteries are the branches derived from the main splenic artery and are eccentric in position. The white pulp appears like the islands in a sea of red pulp because the amount of the percentage of white pulp is very less, so it appears like islands in the sea of red pulp. That is, a vast distribution of red pulp is there, and it makes up around 25% of the splenic tissue, and it is composed of periarterial lymphatic sheath, which is I already mentioned, which is abbreviated popularly as PALS, and it consists of marginal zone and germinal centers. Those things also I have mentioned. And PALS contains mainly the T cells and macrophages, and the marginal zone is made up of predominantly macrophages. It also contains an abundance of blood antigens and thus plays a major role in the immunological activities of the spleen. And the germinal centers are rich in B cells associated with antibody mediated immunity, and the cells are suspended on reticular fibers. Coming to red pulp, as I mentioned earlier, the red pulp makes up around 75% of the splenic tissue, and the red pulp is consisting of venous sinuses filled with blood and cords of splenic tissue called splenic cords of Billroth's. And the Billroth's cords consist of RBCs, macrophages, lymphocytes, plasma cells and granulocytes. And sinusoids are leaky dilated blood vessels with elongated endothelial cells surrounded by incomplete basal lamina. And the spaces between the endothelial cells permit only flexible cells to pass easily from red pulp cords to the lumen of the sinusoids. And the macrophages present in the red pulp cord ingest worn out RBCs, hence the spleen is regarded as the graveyard of RBCs. So this is another diagrammatic view of the section of, that is, transverse section of the spleen, so which is clearly shown in this diagram. Coming to the last part, that is circulation through the spleen. Splenic artery enters the spleen through hilus. Initially it passes through the trabeculae for short distance and branches out as it enters the intertrabecular spaces. And the main branches of splenic artery are called as central arteries. And the central arteries are surrounded by a dense sheath of lymphocytes that constitutes the white pulp. And the straight branches of central artery are called penicilli, and each penicilli show localized thickening of its wall called ellipsoid. And the wall of the ellipsoid consists of concentric lamellae formed by aggregation of reticular cells and macrophages. The lumen of the penicillus is narrow at the ellipsoid. The manner in which blood flows from the arterial capillaries of the red pulp to the interior of the sinusoids is not well understood. According to the concept of a closed splenic circulation, the capillaries open directly into the sinusoids. And according to the concept of open splenic circulation, the penicilli open into the splenic cords and the blood passes through the space between the cells to reach the sinusoids. And from the sinusoids, blood proceeds to the red pulp veins that join together and enter the trabeculae, forming the trabecular veins. And the splenic vein originates from these vessels and emerges from the hilum of the spleen. So here is the schematic representation or the flow chart of the splenic circulation, which is mentioned here as splenic circulation scheme. And as you observe here, the blood flows from splenic artery to central artery and then to penicillus and then to sinusoids, and from sinusoids to red pulp vein and then to trabecular vein and finally to splenic vein. So this is what the importance of the study of circulation in spleen, and with this I have completed the details of the histological structures or the features of spleen. Thank you.